Thank you, Dr. Botstein. Our next speaker, Dr. Solomon Snyder, is also a longtime friend and advisor to the Gruber Foundation, who served for 3 years on its Neuroscience Advisory Board. Dr. Snyder is currently the Distinguished Service Professor of Neuroscience, Pharmacology, and Psychiatry at Johns Hopkins University School of Medicine. Many advances in molecular neuroscience have stemmed from Dr. Snyder's identification of receptors for neurotransmitters and drugs and elucidation of the actions of psychotropic agents. He pioneered the labeling of receptors by reversible ligand binding in the identification of opiate receptors and extended this technique to all the major neurotransmitter receptors in the brain. The application of Dr. Snyder's techniques has enhanced the development of new agents in the pharmaceutical industry by enabling rapid screening of large numbers of candidate drugs. Dr. Snyder received his undergraduate and medical training at Georgetown University. He joined the faculty at Johns Hopkins Medical School in 1966 and established its Department of Neuroscience in 1980. In 2006, the department was renamed the Solomon H. Snyder Department of Neuroscience in his honor. He is also the recipient of numerous other professional honors, including the Albert Lasker Award for Basic Biomedical Research and the National Medal of Science. Please welcome Dr. Solomon Snyder.
Thanks very much, Mary. It's been a pleasure to work with the Gruber Foundation for these number of years since we set up the Neuroscience Award. In contrast to David, who was trained as a geneticist, I, as Mary indicated, went to medical school. I always wanted to be a doctor. I wanted to be a psychiatrist since I was in high school, and I didn't really think that much about science until I sort of stumbled into it, and I only had 2 years at the NIH of formal research training, and so I've always thought that what I should do should have something to do that should be clinically relevant, only there wasn't much to be done. I trained in studying neurotransmitters and drugs, and have continued to study that sort of thing. And when the big breakthroughs in molecular biology came about in cloning genes in the mid-70s, I was already too old to understand it that much. So it was very, very slow in taking advantage. But I followed things very, very much. And in fact, Dr. Botstein's breakthroughs that made the possibility with a polymorphic analysis to find things like the gene for Huntington's disease always fascinated me. And so I'm going to talk to you today just about one project in our laboratory, but it has broad ramifications and deals with Huntington's disease. And this is, let's see if this works. Yeah. Remarkable disease because it just tells us lots of interesting things. As you know, it affects primarily motor function, but then you get generally demented, abnormal movements of the sort that we know in medicine are caused by one part of the brain, the corpus striatum. And indeed, as I'll show you shortly, that part of the brain is affected much, much more than any other part of the brain. And it is indeed a genetic disease, a very specific gene abnormality in which there are these repeats that David mentioned. And in 1993, the specific genetic abnormality was identified due to an extraordinary event in science. Namely, scientists worked together as friendly teams without worrying about who scooped who. And it was all due to this efforts of this wonderful lady, Nancy Wexler, who received the Lasker Award for her efforts bringing together all kinds of scientists to get blood samples and patients with families with Huntington's disease, and they found the abnormal gene in 1993. And as David indicated, today is 16 years later, And while there's been a blood test available for it, I was recently told that only about 5% of people at risk for Huntington's disease take the blood test because there ain't nothing to be done. And actually it raises interesting questions about, even though there is nothing to be done, would you want to know that you have a 50-50 chance, by the way. If one of your parents had the disease, you have a 50-50 chance of getting it. So if you have the blood test and you're clean, wow, that's great. If you have the blood test and you're not clean, then you know someday you're going to get it, and that's a story for another day. And of course the goal, I mean, I'm a psychiatrist. Our goal is to find the genes that cause schizophrenia, manic depressive illness, anxiety, and if you find the gene, then you know the protein that causes it, and then you'll find the cure. But here we are, 16 years later, and there isn't any cure. Let me tell you some of the issues involved. As I mentioned, one part of the brain, the corpus striatum that regulates motor activity, is affected so enormously that, as you can see here, in the patient's brain, it has shrunken remarkably, like 90%, whereas other parts of the brain aren't really affected all that much. The cerebral cortex starts to go and people get demented. The cerebellum can be completely normal, even with the most advanced Huntington's disease. And now, a key fact you should know is that gene, Huntington, codes for a protein called huntingtin, and that protein is expressed everywhere in the body. Its levels are exactly the same throughout the brain, and yet only one part of the brain is massively destroyed. And it's in the liver and the kidney and your small toe. And people with Huntington's disease, they don't have liver disease, they don't have anything, it's very, very peculiar. And so just knowing that you have the abnormal gene hasn't done much. So what's to be done? Well, the story goes back to our work with neurotransmitters. Mary alluded to our studies on neurotransmitter receptors, and another area of interest over the years has been finding novel neurotransmitters, and in the late 1980s, early 1990s, largely due to a wonderful MD PhD student, David Brett, we showed that a gas, nitric oxide, which was known to regulate blood vessels, also is a neurotransmitter in the brain, and we're interested in figuring out how it works, because it's sort of a funny, a little gas. How can it signal if it just sort of free diffuses around? And we found that it's regulated in a unique way. The major excitatory neurotransmitter in the brain, glutamic acid, through its receptors, will trigger calcium entry into cells, which activates the enzyme that makes nitric oxide. And that's how you can have, when nerves fire, calcium rushes into the cell, and that would activate the formation of nitric oxide, and that's how nerve activity can turn on nitric oxide. And as I indicated, it's if nitric oxide, which is a very chemically reactive free radical, just freely diffused around cells to its targets, it would never get anywhere because it would be taken up by a lot of substances like glutathione that would just overwhelm it. So it has to be hand-delivered to its target. So once we had identified the enzyme, we looked for proteins that would bind it, and through the work of a wonderful MD PhD student, Sami Joffre, now on the faculty at Cornell Medical College next door, he said, well, let's look to see if we can find proteins that will bind to it that might be scaffolds, and he discovered a protein that we call capon, a scaffolding protein that connects nitric oxide synthase to other proteins. And one of those was a protein that nobody ever heard about, discovered in the veterinary school at Auburn University, that is called dextras. It's a member of the RAS family of cancer genes, and it's activated by cortisone-like hormones like dexamethasone. We call it dextras. And the nitric oxide is delivered to it by being bound this way, and it changes its function. And in this study by Jamie Chia and Sang-Won Kim, we show that it actually regulates iron entry into cells. Now, because we have been working on this obscure protein dextras, we were aware of the existence of an even more obscure protein called race, identified by my friend Greg Sutcliffe at the Salk Institute 10 years ago, when he was just screening for novel proteins in the brain, things that were expressed in the brain uniquely, or not uniquely. And it's this broad screen, he came up with a protein that didn't look very interesting, so it was so obscure a finding, it was published in a journal that nobody even heard of the name of the journal, far less the name of the protein, Journal of Neuroscience Research, and he called that protein race, because it was part of this RAS family, but there was something very remarkable about this protein. This protein, race, was named RAS homolog enriched in the striatum. That's why they call it race. And here is a picture of, you can see, this is a caudate enormous enrichment, and this stuff in the hippocampus is probably nonspecific. But anyway, enormously and selectively enriched in the corpus striatum. And that caused a postdoctoral fellow in my laboratory, Srini Subramaniam, and myself to say, okay, maybe, how about the following idea why a protein like Huntingtin that's all over the body, all over the brain, could selectively destroy the striatum? Maybe it binds to something unique to the striatum. Let's see if it binds. And protein-protein binding is a very powerful tool of modern molecular biology that we do a real lot in our lab. And Srini showed that sure enough it binds. Now interestingly, the mutant form of Huntingtin binds better than the normal form. This is in transfected cells. This is a cell line, a continuous cell line of neurons just from the corpus striatum developed by Marcy McDonald at Harvard Medical School. And here she has forms of this cell line that express normal Huntingtin or the mutant Huntingtin that has all of these glutamine repeats. And here you see race is binding to mutant Huntingtin very well and much better than to normal Huntingtin. And these are transgenic mice with Huntington's disease, and you can see terrific binding. Now, so that's nice. Does that binding have any function? The function we're looking for is destroying neurons in the corpus striatum to cause Huntington's disease. And sure enough, let's again go back to the same old cells. Let's look at the transfected cells. So you just put in a control. Just put in race, and this is survival. So they're just doing fine if you put in race. If you put in normal Huntingtin, they're doing just fine. If you put in mutant Huntingtin, they're doing just fine. If you put in race together with normal Huntingtin, they're doing just fine. But if you put race in together with the mutant Huntingtin, it kills them. Now let's take Marcy McDonald's striatal cells, and basically you see the same thing. If you put in race with the mutant Huntingtin, it kills them off. Race with the normal Huntingtin, it doesn't kill them off. Let's flip the coin. Instead of putting in race, let's take it away. Well, a neuronal cell type called PC12 cells has race normally. And in this particular cell line, if you can kill them with mutant Huntingtin. Okay, and then we use something called RNA interference to get rid of the race. And we had 4 different forms of the RNA interference probe, and with different efficacies. And actually, number 4 is the only one that actually gets rid of the race. And you get rid of the race, you stop killing the cells. So that looked pretty impressive. And it raised the question, well, how does it actually do that? One interesting line of evidence that had been developed over the years has to do with the mutant Huntingtin forms little aggregates inside of cells in the cytoplasm and in the nucleus. And people originally thought that the aggregates are bad. You have a piece of junk clotting, and that's bad for you. Actually, clots aren't so bad because they heal your... So I cut you, you get clot, you get better. So maybe the clots are good for getting rid of, maybe soluble mutant huntingtin kills you, and you aggregate it so it won't be soluble, so it will be just sequestered away. And in fact, indeed, more and more evidence indicates that that's the case. In fact, indeed, if you look at the corpus striatum of patients' brains, they have mostly soluble huntingtin. If you look at the cerebellum, where you don't get any destruction, they have largely the aggregated form. So let's see what happens with race. Actually, race decreases the amount of aggregates and increases the amount of soluble materials. Okay, so that is interesting. Now, the question is, how does that all come to be, and how does it actually work? Well, let's go back to what is race and how does race do its thing. Race is in the family of what they call small G proteins, which bind the nucleotide GTP and diddle around with it. And it's not important that you know that in detail, but binding GTP and acting upon it is a classical way whereby proteins of this family, of the G protein form, work. So the immediate question is, how does race do this killing? Does it kill through its G protein function, which involves binding GTP? And that's easy to study because you can make mutant forms that will not have the GTP binding by mutating serine 33. Now, remember, if you get rid of aggregation, then you're going to kill. So here we put in our race, and we get less aggregation. Here's your aggregate, and here the aggregation is decreased, and we kill. Now, let's abolish the G protein function of race by mutating serine 33 to arginine. You still get rid of the aggregation, and you still kill. Aha! Okay, so the classical G protein function has nothing to do with killing the cells. What might? Well, about 4 or 5 years ago, a very nice study by Joan Steffen and a nice team, that's very good people working on Huntington's disease, they found an interesting modification of this protein huntingtin that consists of adding a sumo group, which is like adding a ubiquitin group, which is another small protein. And it's just a modification of proteins, which people don't really know exactly what it does, but it does a lot of different things, and it's not altogether pinned down. And what they showed is that when the protein huntingtin has this sumo modification, it becomes more soluble. It stops aggregating, and it kills. You actually, even in a fly model of Huntington's disease, you have the sumo modification, and you kill neurons. So we said, if the race protein isn't killing neurons through its G protein function, maybe it's killing neurons through this sumo business. But of course, we don't know that race has anything to do with the sumo. All we know is that if you put the sumo protein onto Huntington, then that will help Huntington kill neurons. And so the question is, does race have anything to do with putting the sumo group onto the Huntington protein? And so Srini looked, and sure enough, it does. This thing shows that it's putting the sumo group, here's race, with the mutant Huntington, and it puts the sumo group onto it. Now, here you see it putting the sumo group onto it and killing neurons. Now, the sumo group is put onto the amino acids lysine, and there are these 4 lysines that get the sumo group. So if we want to stop putting the sumo group on, we just mutate the 4 lysines, which we do over here in the last column, and we don't get any more aggregation. No more sumo, no more aggregation, and no more killing. you've got to put the sumo group to kill, which we knew already, but it's race that's putting the sumo group on. And how is race going about putting that sumo group on? Well, I'll show you a little bit about the process of sumo relation. There are a couple of steps. There's the E1, the E2, and the E3 step, and don't worry about them in detail. But you notice the E2 is also called UBC9, and the key thing is the first two steps happen with every single target for sumo relation. The third step is where you have a whole series of enzymes that put on the sumo group. These are all called E3 type agents, and they have to also interact with UBC9 to do this. So could race be an E3 ligase for the sumo relation? And the answer is indeed. It does indeed interact with UBC9. It does indeed put on the race, puts on the sumo group, and if you get rid of those lysines, it won't put on the sumo group. And so then the question is, okay, how do we prove that putting on that sumo group by race, that race we know can put the sumo group onto Huntington? And now the question is, how do we know that race putting the sumo group onto Huntington is what kills neurons? And the answer is, let's get rid of sumo, which we can do by a technique called RNA interference. And if you, here we are killing with race and mutant Huntington, and we get rid of the sumo, and we stop killing. So the bottom line is very simple, that now we have interesting insights. So we talk about the wonders of genetics. You find the abnormal gene, it doesn't necessarily solve the problem of why, in this case, mutant Huntington kills neurons and destroys uniquely one part of the brain, the corpus striatum, predominantly. The answer is, the way it works is this mutant Huntington, which is everywhere, but in the striatum, it interacts with this protein race, which puts the sumo group on it, which disaggregates it, which makes it toxic, kills neurons. And if you think about it, this could have notable implications. Obviously, if we stopped race from binding to mutant Huntington, maybe you would prevent the development of the neurotoxicity and the clinical symptoms. And we're already screening with collaborators for drugs that will block that binding or block the adding of the sumo group. And to actually test that, we already have mice that don't have the race. The gene for race has been knocked out, and we're mating them with mice that have the mutation that causes Huntington's disease. And it'll take better part of a year to have an answer from there. But meanwhile, we're actually looking for drugs. So I thought I'd give you this idea of how one can actually go from very, very basic neuroscience and try to take genetic discoveries and place them through ordinary old-fashioned biochemistry into diseases and point a way toward developing the therapeutic agents. And my job is talking to you. The real science is done by people in the lab. In this particular case, one postdoctoral fellow, Srini Subramanian, gets all the credit. And we work on a lot of other things in our lab. I told you about nitric oxide, which is the focus of Bala Selvakumar in the lab. And gases as neurotransmitters are a fascinating area, and we're looking at other gases. The most recent one is hydrogen sulfide. It makes eggs, rotten egg smell, which it turns out we had a paper in Science last year showing that regulation of blood vessels is actually more by hydrogen sulfide than by nitric oxide. Work done by Asif Mustafa and Mo Gatala, and we work with inositol phosphate that other people like Michael Klodowski work on. And to have consistency, since the boys and the girls who are the students come and go, Adele Snowman graduated from college in 1972 and joined my lab, and just had so much energy. She would be in at 5:30 in the morning, work till 9:30 at night, because she was working together with graduate students who were doing this ligand binding that Mary alluded to. The work on the opiate receptor wouldn't have been possible without Adele, who is still our lab manager, and it still works amazingly. And tissue culture is done by Linda Hester, who joined the lab in 1977, and it's a wonderful group of boys and girls, which is why I like to wake up in the morning and come to the lab. And thank you. Sure. Linda wants to ask a question. Let me turn the lights on.
So, Huntington's, like many neurodegenerative diseases, occurs later in life. Do you have any insight into the late onset of Huntington's?
Right. Not really. For all kinds of diseases that have a late onset, there are standard hand-waving explanations, like, well, these things are ongoing, and then as you get other insults with aging, for instance, in Alzheimer's disease, there's a clear accentuation by atherosclerotic changes. And so they say, well, you got a little hardening of the arteries, and then the Alzheimer's changes start kicking in and they work together. And so I could give you 25 other explanations, which are worth about as much as you're paying for them. So I have a feeling there may well be a good, rigorous answer to the question, which would be important for Parkinson's and Alzheimer's and Huntington's, and I don't think any of us know. Yes?
Were there any effects in the mice that had race knocked out?
I didn't hear the question.
Were there any adverse effects in the mice that had race knocked out?
Oh, a question. In the mice with race knocked out, what do you see? Well, they haven't been all that well characterized, but thus far we don't see very, very much. Our colleagues in Italy who actually made the mice for us have done some characterization. And there's a little bit dopamine. The dopamine is a neurotransmitter of the corpus striatum, and there are a few abnormalities, but nothing to write home about. And if they look very carefully, their motor activity is a little changed, but also nothing very bad. In our hands, they run around and they do just okay. Which is actually good, because then we can use them for breeding studies, things like that. Okay, thank you very, very much.